Hello everyone. I'd like to thank the ANA for inviting me to speak at their annual conference this year. The topic of our talk today is the genomics of vitamin D, and specifically as it relates to immune function. I hope to make it fun and informative, particularly given our current context of COVID-19. I'd like to disclose that I'm actually a scientific consultant of the DNA company, a shareholder, and I receive a monthly stipend from them. However, the topic and the content for today's lecture is entirely derived from the academic literature. After participating in today's lecture, we should be able to explain the multi-step, multi-organ nature of vitamin D production, really appreciating that it is a cascade. We should be able to identify common misconceptions about the various forms of vitamin D, recognize the hormonal, this is going to be key, recognize the hormonal nature of vitamin D and its role via the VDR or vitamin D receptor in modulating gene expression. This is the central theme for today's talk. We should then be able to describe the impact of inter-individual genomic variations at each step in the production, transportation, and activation of vitamin D, i.e. in each step of that multi-step cascade. We should be able to examine the role of inter-individual genomic variations in vitamin D utilization as it relates to immune resilience. I'd like to establish a little historical context for our talk today. I think one, it's just fascinating, but two, it really helps to emphasize how we've matured in our understanding of what vitamin D is and how and where it's linked to the overall bigger picture of nutrition. The discovery of vitamin A by McCollum and Davis just over 100 years ago in 1913 ushered in the era of vitamins and the appreciation of specific dietary micronutrients and cofactors essential for optimal health. In other words, it was just over 100 years ago that we began to appreciate that beyond macronutrients, the proteins and carbs and fats that we were eating, that there were microcomponents, there were these other factors in our diet that were they not to be there, or were they to be there in unhealthy levels or unhealthy balance levels, we would have consequences, we would have ill health. And this is when we ushered in and we started naming these alphabet letter vitamins, of course the first one being designated vitamin A. Now just a few years after the discovery of vitamin A, i.e. just under 100 years ago, following in the footsteps of McCollum and colleagues, Sir Edward Millenby of Great Britain attempted to address the root causes of rickets, the disease of early childhood development that leads to poor bone density and the bowing of the lower limbs, the bowing of the lower bones in the legs, which was highly prevalent in Scotland at the time. Mimicking the Scottish diet, Sir Millenby fed an oat-rich diet. The oat-rich diet was prominent in the Scottish diet, to dogs. But inadvertently, during this experiment, he kept the dogs indoors and away from sunlight. Before long, remarkably, he described a phenotype in these dogs that was identical to rickets in human beings. Now, Sir Millenby found that he could cure the disease by administering cod liver oil. He, however, coming on the footsteps on the heels of vitamin A's discovery, he made the incorrect assumption that the curative micronutrient in the cod liver oil was vitamin A. Ironically, it was actually McCollum and colleagues who once again corrected that misconception, and they concluded that the factor in cod liver oil that could ameliorate the signs of rickets was actually not vitamin A, but a new, and because they were in the world of discovering vitamins, they named it this new vitamin, vitamin D. So let's have some fun. This is likely going to be the most important slide in the presentation, or one of the most important slides in the presentation. We know that we make vitamin D, as we said earlier, in the dermis, the epidermis actually, wherein we're going to convert 7-dehydrocholesterol into the cholecalciferol. And how are we doing this? Well, we're doing this through a photosynthetic reaction, i.e. the UVB fraction of sunlight exposure is what converts that 7-dehydrocholesterol into cholecalciferol. What we're going to do now is we're going to start layering important enzymes, important genes, the genes that encode important enzymes, to highlight the multi-step cascade nature of vitamin D synthesis, and thereafter its activation into its hormone, and thereafter its activation of its hormonal receptor, and so on and so forth. So the first of the things we want to highlight is the HAL enzyme and the HAL gene. So there's this gene, the HAL gene, that produces the HAL enzyme. And what does this enzyme do? It produces transuronic acid. What is transuronic acid in the epidermis? It acts as a natural sunblock. So to the degree that you produce this transuronic acid is the degree that you block UVB. So the activity, which is genetically determined, there are different genotypes, different versions of the HAL gene, making different versions of the HAL enzyme. There's marked inter-individual differences to the levels of transuronic acid that we produce, i.e., we have marked inter-individual differences to our internal sunblock, and that internal sunblock is going to diminish the impact of the UVB, which is of course going to diminishing impact the conversion of 7-dehydrocholesterol into cholecalciferol. But it doesn't end there, not by a margin. We've also got this other enzyme, this other gene encoding another enzyme. The DHCR7 stands for dehydrocholesterol reductase, or the 7-dehydrocholesterol reductase. And the name of the enzyme tells you what it's doing. It is reducing the 7-dehydrocholesterol into cholesterol. So yes, in fact, look at how intimately related the whole cascade of vitamin D is to cholesterol. Now mind you, this is the cholesterol being produced in the skin. So you see, here's the point. This gene, the DHCR7 gene, again, has different genotypes. We have as individuals different versions of that gene, different levels of DHCR activity. Individuals with a greater degree of DHCR activity, because the 7-dehydrocholesterol is at the crossroads, either going to be reduced by this DHCR into cholesterol or be converted into D3, we could see that different genotypes, whether you have the faster DHCR7 gene, where you shunt more of your 7-dehydrocholesterol into cholesterol away from D3, or vice versa. Well, let's continue. Other than, of course, producing D3 through sunlight exposure, and that's a really important consideration in the months that we're going in here in the northern latitudes, we know that we can also ingest orally, either through foods or through supplementation, D3. And the body has an absolute way of absorbing orally, intestinally delivered D3, and this is through the chylomicrons, again, directly, intimately related to the lipid transportation system of the body. And through these chylomicrons and via the thoracic lymph duct, we are going to absorb the orally administered D3 into the bloodstream. Now, once in the bloodstream, either made in the skin or orally ingested, we've got this important protein known as the VDBP. And the VDBP is the binding protein, vitamin D binding protein. It's the taxi of the D3. Once that D3 gets into the bloodstream, it has this all-important taxicab that it must bind to, essentially, to be transported. And that VDBP, that vitamin D taxicab, by the way, it's the product of a gene. And there are, you guessed it, versions of that gene, which means we have different levels of vitamin D3 transportation efficiency. Now, the VDBP is going to take the vitamin D3 that has been made or ingested, take it to the liver, wherein another enzyme, the CYP2R1 enzyme, is going to biotransform the D3 into 25-hydroxy-D. 25-hydroxy-D now becoming the primary circulating form of the vitamin D. Importantly, this enzyme, CYP2R1, is magnesium dependent. Cannot stress this enough. It's one of the reasons why it's so ultra important to have healthy levels of magnesium in our diet. Now, once we've made the 25-hydroxy, this is the pro-hormone. The 25-hydroxy-D, is that the end and be-all? By no stretch of the imagination. We're going to one more thing. We not only can ingest D3, we can ingest D2, which of course was the plant-derived or the ergocalciferol. It too will be absorbed via the same chylomicron mechanism. It too will be transported by the vitamin D binding protein to the liver, and it too will be converted into 25-hydroxy-D. So many times I feel and I've heard there are misconceptions about what D2 and D3 really are. Many clinicians, surprisingly, I hear them thinking that D3 is quote-unquote the activated and the end and be-all of vitamin D, and they somehow give preference of it. We're not going to get into the differences or preferences between D2 and D3, but just highlighting that both D2 and D3 will make it to the liver according to your vitamin D binding protein, wherein they will both be activated into 25-hydroxy-D. Now, thereafter, again, the VDBP will grab that 25-hydroxy-D, take it to the kidney, and in the proximal convoluted tubules of the kidney, we've got yet another of these CYP enzymes, CYP27B1, more commonly known as the 1-alpha hydroxylase. And that again is magnesium dependent, and that enzyme completes the biotransformation of the 25-hydroxy-D into its final hormonal form, the 1,25-dihydroxy-D. That 1,25-dihydroxy-D, having been its evolution completed in the kidneys, further again is transported by the vitamin D binding proteins. It gets into the bloodstream. The VDBP now transports that hormone to all of the cells of our body. That hormone enters our cells and it activates its receptor, the vitamin D receptor, which is yet again the product of a gene, which is yet again variable. There are different versions of that gene that we might have as individuals. There's marked inter-individual variances to the binding capacity of the vitamin D receptor. And once that 1,25-dihydroxy vitamin D binds to VDR, it activates the receptor, and that hormonal receptor does what all hormonal receptors do. It becomes a DNA transcriptional regulator. Let's repeat that slowly. Hormonal receptors, once activated by their ligands, how does the whole hormonal system work? It works by becoming activated by its ligand, by its hormone, becoming a transcriptional regulator, entering into our cells and eliciting profound gene expression regulation. Now there's some nuances here that we're going to get into later, and that is that once the vitamin D receptor is activated, it actually then couples with another receptor, the RXR or retinoid receptor, to form a heterodimer, and it's actually the heterodimer that is what binds to our genes and elicits the gene expression changes. We'll get into this in more detail later on. Okay, so let's extrapolate a little further on the concept of the VDR and its initiation of gene expression to create what we call the vitamin D or the VDR transcriptome. The VDR, the vitamin D receptor, as we've noted, is present in the nucleus. Now, in different hormonal systems, the receptors might be present embedded in the cell membrane, such that that receptor has an outward-facing binding region, such that the hormones circulating in the bloodstream bind to their receptors embedded in the cell membrane, and thereafter the receptor dislocates from the cell membrane, travels into the cytoplasm, from the cytoplasm travels into the nucleus, and does the job of gene expression, modulating gene expression. In the case of the VDR is already in the nucleus, and it's in the nucleus present in an inactive form. Now, when that VDR is present in its inactive form, in other words, in the nucleus, when it has not yet been bound by the 1,25-dihydroxy-D, it's not yet been activated, what you find is that VDR suppresses gene expression. It suppresses the genes that have the vitamin D receptor binding regions. Once it becomes activated, mind you, with the coupling of the VDR and the RXR, the retinoid receptor, that's when we induce gene expression. So what do we have? We've got that cascade where we converted the D3 into the 25-hydroxy, the primary circulating form, the prohormone in the liver, produced in the liver. The 25-hydroxy activates in the kidney into the 1,25-dihydroxy. Now that's in the bloodstream. The 1,25-dihydroxy enters the cells accordingly. And in those cells that have, mind you, the VDR in their nuclei, the VDR is going to be activated upon binding by the 1,25-dihydroxy. vitamin D. It couples with the RXR to form this heterodimer, and it induces gene expression. The modulation between the genes that were being silenced when the VDR is not activated versus when the VDR is activated, this is what determines the vitamin D transcriptome, or more accurately, the vitamin D receptor transcriptome. Okay? And this transcriptome is extremely cell type specific, and it refers to the pivotal cascade of gene expression changes, both the upregulation and downregulation in a cell type specific manner caused by this VDR. What are the noteworthy characteristics that once the VDR transcriptome has been initiated? Well, the genes that are regulated through the activation of the VDR are radically important in cell differentiation. Literally, in the very early stages of embryonic development, when our stem cells, when those undifferentiated cells are now taking on unique functions, those unique functions, that cell differentiation, a lot of it is actually driven by the activation of the VDR. Moreover, as we continue as adults, as throughout our lives, the differentiation of stem cell populations rely to a large extent on the activation of the VDR. The activation of the VDR is important in several layers of the immune system, and it accounts for the regulation and modulation of over 1,000 genes. A bit more of this to come later. Now let's shift gears just a little. You see, at about the same time that McCollum and colleagues and Sir Edward Millenby were unraveling the mysteries of the newly discovered vitamin D, another British physician, Sir Alexander Fleming, was making an equally intriguing discovery. He discovered, as much as we know him more for antibiotics, he discovered the first antimicrobial peptide, or AMP, which of course is lysozyme. So while Sir Fleming is often more famously known for his discovery of penicillin, and likely because of this latter discovery, his initial work on AMPs was overshadowed and did not receive the attention it deserved until much more recently, actually, where AMPs have now come back into the limelight because of their radically important role in the innate immune system. Importantly, the pleiotropic functions of AMPs extend well beyond antimicrobial activity and include regulation of inflammation, angiogenesis, and wound healing. And by this fact, because of this, and because of the pleiotropic properties of AMPs, alternate names to emphasize their functions beyond antimicrobial activity, such as host defense peptides, alarmins, and other names, have actually been suggested. Remarkably, after that first discovery by Sir Fleming of lysozyme, to date over 100 AMP genes and their encoded peptides have been identified from a variety of tissues and epithelial surfaces, including skin, eyes, ears, buccal lining, gastrointestinal tract, respiratory tract, lung epithelia, immune, nervous system, and urinary systems. Essentially, every border of entry into the human body, those cells that act as those border barrier function cells, express these AMPs. And of course, why shouldn't they? Because these AMPs are part of our innate immune response, the first line defenders to our body. These AMP genes and their peptides they encode are typically quite small. The peptides vary from 10 to 150 amino acids. These are not very large proteins at all. The sequence diversity of human AMPs, these 100 plus genes and their various sequences, enable them to adopt various 3D structures capable of attacking pathogens via a range of mechanisms. I'm not going to read out all of these, but we have things like alpha defensins can assemble on the bacterial surface into nanonets to entangle bacteria. We've got that well-defined lysozyme, which is capable of actually cleaving polysaccharides, leading to the lysis of the microbial, the bacteria in question, and so on and so forth. So why do we take this segue to AMPs? And of course, because they're very important in the immune system. Well, what's this link between AMPs and vitamin D, or more specifically, AMPs and 1,25-dihydroxy vitamin D, or even more specifically, AMPs and 1,25-dihydroxy activated VDR? Well, you guessed it. AMP genes, those 100 plus genes that are making these radically important peptides in our immune system, they're part of the vitamin D transcriptome. They're that are going to be regulated once that vitamin D receptor has been turned on. Let's extrapolate a little the concept of the VDR and its transcriptome, i.e. its transcriptional circuitry. The VDR regulates transcriptional programs that control cell growth and differentiation, as we've mentioned. It's radically important in the adaptation of cells, in the differentiation of our stem cells throughout life. It exerts its transcriptional control by binding in a variegated but tightly cell type specific manner, and we're going to pictorially extrapolate on this shortly. By doing what? By binding to these sequences, these vitamin D binding elements, or more accurately, frankly, vitamin D receptor binding elements throughout the genome. Okay? And these elements, these sequences that are scattered throughout the genome, then once the activated VDR binds to them, the gene in question is regulated, its expression is regulated. The cell type specificity of the VDR transcriptome, is accomplished in a genius manner. Now you see, thus far we've been saying that you've got the 1,25-dihydroxy vitamin D, it binds to the VDR, it activates it, the VDR forms a heterodimer with the RXR, the retinoid receptor. But you see, remarkably, and again we'll show this pictorially, we then further couple that heterodimer with very cell type specific cofactors that would be unique to the cardiac cells that the 1,25-dihydroxy vitamin D has entered into, to the neural cells, to the B cells, the T cells, and so on and so forth. So different cell types in the nuclei of different cell types, other cofactors will be contributed to the heterodimer such that that heterodimer becomes a cell type specific transcriptional regulator. And we'll see examples of this. I really want to emphasize that to date and counting, mind you, there are over 2,200 vitamin D receptor binding regions that have been discovered throughout the genome, accounting for a control. The vitamin D, the VDR transcriptome, is estimated, as we've said, to be over 1,000 genes. Let's see how this works pictorially. Well, a picture paints 1,000 words, so let's look at how this VDR is actually activated and how it accomplishes that genius cell type specific gene regulation. We start with the 1,25-dihydroxy vitamin D binding to that inactive VDR, which is already present in the nucleus. That leads, of course, to the activated VDR in the nuclei of the cell. So at this point in time, you've got your 125, the hormonal 125-dihydroxy vitamin D circulating in your bloodstream after you've gone on that nice stroll in the sun, or of course having taken the vitamin D3 supplementally or eaten some of that fish that you got your vitamin D3 from. It had become activated in the liver into its prohormone. It then further became completed via the kidney into the 125-dihydroxy D. The 125-dihydroxy D enters all of the cells of the body accordingly, finds the cells, and the cells that have the VDR in the nuclei, we now have the activated VDR. Okay, this is fairly homogeneous. How does the cell specificity come about? Well, we now then, activated VDR binds to the RXR. That gives you the activated heterodimer, or the VDR RXR. This is where the genius comes in. You see, in each of the different cells, those cardiac cells versus neuronal cells versus muscle cells versus B cells and T cells and so on and so forth, each of those cells, as much as the activated VDR RXR is homogeneous in all of these cells, what is unique is that each of those cells provide cell type specific transcriptional cofactors. So what you then really end up having is you end up creating all of these cell type specific VDR complexes, the VDR RXR complex. Those cell type specific VDR RXR complexes, they then initiate transcriptomic activity that is unique to the B cell, unique to the T cell, the cardiac cell, the neuronal cell, the muscle cell, and so on and so forth, which of course is precisely the point, that through the homogeneous presence of 125-dihydroxy D, creating a homogeneous activation of the VDR, we get not a homogeneous, but we get a very heterogeneous, cell-specific cellular behavioral change by the activation of the VDR. It's really an incredibly genius system. So now that we've seen this frankly miraculous way in which that one thing, that one hormone can elicit cell-specific, and by the way, this is not unique to vitamin D. All hormones, through their receptors, the same concept plays out, which is why estrogens can affect, in our bodies, different cells in different ways. It's the same estrogen, binding via the same estrogen receptor, but the different cells then contribute other cofactors that determine the transcriptome associated with estrogen in our unique cells, associated with androgens in the androgen receptor, associated with insulin in the insulin receptor, and so on and so forth. So now we've activated the VDR, and it's doing its job of creating a transcriptome. It's doing its job of activating gene expression in different cell types. What are some of the salient features that occur? How does the biology of cells change, especially as it relates to immune and inflammatory responses? I'm not going to go through every point here, but I want to highlight a couple of things. The 1,25-dihydroxy deactivation of the VDR stimulates the differentiation of monocytes into mature macrophages and induces macrophage chemotactic ability and phagocytosis. So in other words, right off the bat, that VDR activation, radically important in one of the primary hallmarks of our innate immune system. The VDR activation suppresses Th1 proliferation with concomitant reduction in interferon gamma, which is pro-inflammatory, needed, but pro-inflammatory. So in other words, vitamin D and its hormonal activation and its VDR activation suppress aspects of the more inflammatory components of our immune response. Specifically and remarkably, VDR activation polarizes the adaptive immune response by orchestrating the appropriate transcriptomic differentiation, shifting from a Th1 dominance response to a Th2 dominance response. And that's really important because in so doing, when we shift from the more inflammatory Th1 response to the Th2 response, what is happening here? We're suppressing the production, we're suppressing the progression, I should say, of inflammation through reducing the generation of inflammatory cytokines like IL-6 and its brothers and sisters, i.e., by shifting from the Th1 to the Th2 modality of the immune response. Now let's revisit that amazing cascade that we went through and simply highlight the genes with their enzymes involved at different steps of the cascade for which we've now discovered variations in those genes so that we can see the marked opportunity for inter-individual differences in this cascade, which means what? Ultimately, it means innate, genetically predetermined, innate differences to the activation of our VDR, which means we have innate, genetically predetermined or predisposed differences with which we can activate the VDR transcriptome. And of course, as we can see, that's going to then relate to innate immune resilience differences. So here we go. We've got the HAL gene and its enzyme and variations within it. We've got the DHCR gene and its enzymes and variations within it. We've got the CYP2R1. We've got variations in the VDBP protein. We've got variations in the VDR itself. The very gene that makes the VDR is variable one person to the next. We've even got differences in the RXR gene. How does this play out? Let's see how this plays out. This is a perfect example of what we call functional genomics. And this is the emphasis. Everything that we've said leads up the drum roll to those genes that we mentioned in the cascade. And here are examples in these genes. Those RS numbers are SNPs. They are examples within these genes that there are variations. Those variations, those SNPs, lead to different alleles. So for example, let's take the VDR. In the VDR, there's a very well described SNP, the RS15-44410 SNP, and that SNP defines 3 different genotypes. Some individuals may have a CC genotype, homozygote CC. They may have a heterozygote CT or a homozygote TT genotype. And depending on their genotype for that SNP, the binding capacity of the receptor, the very end point where that receptor is now going to be activated, the efficiency with which it can be activated dramatically differs one person to the next. And similarly, there are variations throughout all of the other genes. So that we see here functional genomics at work. This is the bottom line of it, that we can actually remarkably predict where is a person in this cascade, what does their innate genetic makeup for each of these genes tell us about the efficiency, their innate efficiency of the vitamin D production, evolution cascade, activation cascade, ultimately leading to the activation of the VDR, ultimately leading to the activation of the VDR transcriptome, and thereafter the impact that has on their immune function.